Welcome to today's exploration of the revolutionary world of next-generation sequencing, a technology that has completely transformed genome analysis. Imagine just a few decades ago, the idea of reading an entire human genome seemed like a far-off dream. But today, thanks to next-generation sequencing, it's entirely possible to sequence an entire human genome in a single day. This has opened up new frontiers in our understanding of life at its most fundamental level. From unlocking the mysteries of genetic diseases, to helping us understand our evolutionary past, to even finding new ways to feed our growing population, next-generation sequencing is making what was once thought impossible, possible. In this video, we'll be delving into the intricate process of next-generation sequencing, taking you step by step through this remarkable technology that's reshaping our world. Now, let's dive into the intricate process that powers this revolutionary technology. The first step in next-generation sequencing is sample preparation. This process starts with the isolation of DNA, our blueprint of life. Once we have a purified DNA sample, our next goal is to generate smaller, more manageable fragments. This fragmentation process enables us to analyze the DNA more efficiently. With our DNA now in bite-sized pieces, we can move on to the exciting part, ligation. Each fragment of DNA is ligated or attached to specific oligonucleotides. These oligonucleotides serve two important roles. First, they act as sequencing binding sites, providing a platform for the sequencing process to take place. Second, they include an additional complementary sequence. This sequence is designed to hybridize or bind to the flow cell, a critical component in the next stage of our journey. Once our DNA fragments are prepared and ligated, we're ready to move on to the next step. With our DNA fragments prepared, we're now ready for DNA amplification and bridge building. Our journey begins with DNA amplification, a process carried out by the workhorse of molecular biology, the polymerase chain reaction, or PCR. This mechanism synthesizes a complementary sequence to our single-stranded DNA fragment. It's like creating a mirror image, an exact replica. This process happens simultaneously for all DNA fragments on the flow cell, creating a bustling city of DNA replication. But what happens next? The DNA is denatured, and we're left with the single strand that's still attached to the flow cell's oligonucleotides. The other strand, the unattached one, is washed away, like a ship sailing off into the distance. Now, we move on to an intriguing process called bridge building. It's not about constructing physical bridges, but molecular ones. You see, we have two types of oligonucleotides on our flow cell. The second type, which until now has been waiting in the wings, steps forward. It hybridizes or binds to one of the sticky oligonucleotides on the plate, building a bridge between them. This bridge building sets the stage for another round of amplification. Again, a polymerase synthesizes the complementary sequence, like a dedicated artist meticulously crafting a masterpiece. This whole process from bridge building to amplification is aptly named bridge amplification. It doesn't stop there. The DNA is denatured once more, but this time, both fragments stick to the plate. The process repeats, building bridges and amplifying again and again, like an orchestrated molecular dance. After several rounds of this process, what do we have? From one DNA fragment, we've generated multiple copies through PCR. We have reversed strands, we have forward strands, a veritable army of DNA copies, all from that initial single fragment. Now, one of these strands is cleaved or cut, and we're ready for the next step. After several rounds of this process, we have multiple copies of our DNA fragment ready for sequencing. Now that we have our amplified DNA fragments, we can begin the sequencing process. This intricate process involves the use of fluorescently labeled nucleotides, each color representing one of the 4 nucleotides that make up our DNA: adenine, guanine, cytosine, and thymine. Think of these as the building blocks of life, each with its own unique color code. Let's narrow down our focus to just one of these amplified DNA fragments. A primer binds to the sequencing binding site, ready to kickstart the sequencing. The magic begins when these fluorescently labeled nucleotides join the party. Only one will fit at a time, and when it does, it lights up. A laser excites the fluorescent label, resulting in a light signal. This signal is captured and recorded. For instance, if the blue light flashes, we know we've just added a thymine to our sequence. This process repeats, one nucleotide at a time, slowly building up the sequence. But remember, we're not just sequencing one fragment. We're sequencing millions at the same time, all of them sending out light signals simultaneously. It's like a magnificent microscopic light show, all happening on a tiny flow cell. Now, sequencing is just one part of the story. The other crucial component is data analysis. After all, what good is a light show if we don't know what it means? This is where the power of informatics tools comes into play. They capture these millions of light signals, or reads, and start making sense of them. They overlay these reads, aligning them to reference sequences, identifying variations, and piecing together the puzzle that is the genome. This process, from preparing the sample to sequencing the DNA and analyzing the data, is an intricate dance of science and technology. Each step, each process, is integral to unlocking the secrets of our DNA. And that's how next-generation sequencing works, from sample preparation to data analysis. So, we've walked through the entire process of next-generation sequencing. We started with the revolutionary impact of this technology, how it has transformed genome analysis, making it possible to sequence an entire human genome in just a single day. We dived into the intricate steps of the process, starting with sample preparation and fragmentation, followed by DNA amplification and bridge building. We then explored the fascinating world of sequencing and how fluorescently labeled nucleotides, excited by a laser, give us the sequence. Lastly, we touched on the crucial role of data analysis. It's an essential step as it helps us make sense of the millions and billions of reads generated by next-gen sequencing. This is where we overlay these reads, using informatics tools, to create a complete picture. With next-generation sequencing, we're unlocking the secrets of the genome, paving the way for groundbreaking discoveries in science and medicine. Thank you for joining us on this journey into the world of next-generation sequencing.